Welcome back to the Health Longevity Secret Show with Dr. Robert Lufkin. DNA epigenetic methylation clocks are becoming the gold standard in longevity for determining biological as opposed to chronological age. Today, we are speaking with Dr. Yelena Budovskaya, CEO of Trume Labs, that makes a consumer epigenetic methylation clock for $99. Yelena obtained her Ph.D. in molecular genetics at Ohio State University and then studied the genetics of aging at Stanford University, leading her own research program in aging and the effects of genetics, epigenetics, and the microbiome on the aging process at the University of Amsterdam. Health Longevity Secret Show listeners who wish to try the Trume DNA methylation clock on themselves can receive a 20% discount by entering the code Dr. Lufkin, all caps, no space, at the trumelabs.com website, and the link will be in the show notes. Now, please enjoy this interview with Dr. Yelena Budovskaya. Yelena, welcome to the show.
Thank you so much for having me.
I can't wait to dive into DNA epigenetic methylation clocks and hear about all the work you're doing and the company as well. But before we do that, maybe you could take a moment and tell a little bit about how you came to be so interested in this fascinating area.
Well, it's actually did not start in very early age. Although I would start wondering about why do we age, like really, I guess about age of 14 or so. And the main reason why was because my grandfather at the moment passed away at the age of 65. And I was thinking back then, my parents were about 30 something, 32, and it sounded like way too close. So I started, and of course, as a young person, you always think your parents going to live forever. Your grandma is always going to be there. Your grandpa is always going to tell you stories and things like that. And that really shocked me. And I was wondering, like, why did he, not entirely why he died, but why did his last maybe 5 years of his life was so miserable? And 65 was very young. And then I've learned that my other grandfather on my father's side actually died at the age of 55. And back then I grew up in Soviet Union, so nobody thought twice about this because we won a big war, and a lot of men went to war and they came in injured, and a lot of people just were happy to have as many years as they possibly can get from that generation. So to me, that was kind of shocking. And as I was interested in biology already then, I was wondering, like, why do we have to age? Why does it even have to start in the first place? And then why for such a developed country, and I always considered Russia to be a developed country, we live so short compared to other Western world. And of course, at first I didn't think about diet. We were going through transition and all the kind of crash of Soviet Union and perestroika and everything. It was not about that. It was just, I thought it was genetics. I honestly thought it was genetic program. So I went to university, I studied biochemistry, actually not genetics quite. I started doing my PhD, and I noticed that everybody's looking at what's cancer is. So how to cure cancer, how to cure diseases. But indeed, aging is actually sort of a cause for most of the diseases that we're trying to cure. So I thought maybe it's worthwhile to look at the cause. So if we can cure aging, we can cure the diseases. So one kind of bird with one shot, or what the phrase is, I don't forget. So that's why I joined, and I actually purposefully went to study this process with Stuart Kim, because he was doing, at that time, he was one of the few people who was using genomics and studying aging. So I thought, I want to look at everything and how the whole process changes when an animal goes from young to old. And that's probably the kind of where the first interest started at the clocks in the particular. Yeah.
Oh, interesting. So clocks. Let's talk about clocks. Clocks are becoming fundamental for longevity research. And can you tell us what is a clock and why are they so important for longevity research?
Well, before clocks, maybe I can do two sentences about why we even started searching and digging that area. So if you think about any disease or any process you're studying, you always need a marker that you measure. So if you go for the physical exam, the doctors measure glucose level, triglycerides, and it's kind of like picture of your physiology, right? Do you have diabetes? Do you have high cholesterol? Do you have inflammation? They might measure CRP. So, and then they can correct it. We cannot correct it unless we measure. And for a long time, the only measurement of your longevity was how long would you live, your maximal median lifespan, which we have to pretty much let you live and wait until you die. So in the human population, one experiment would be a lifetime of one individual, right? So you will never put more than one experiment, and you probably will not, and you have to guess how to split the group, which one is a healthy aging cohort and which one is not a healthy aging cohort. So it was clear to me, if I ever want to do experiment in human, I need a biomarker of aging. And then we learned about all the biomarkers in blood. So the same story, we counted all this red blood cells, white blood cells, every kind of some markers for liver function, kidney function. And we have this physiological clock that you can do, like, I think you can put pheno-age measures like 10 biomarkers, and based on that predicts biological age.
Yeah,
But I
that's
think,
oh, excuse me, that's PhenoAge from Morgan Levine, right?
yes,
And
yes,
we'll
yeah.
put a link to that in our show notes for our audience, and they can enter those values and calculate the PhenoAge.
Yeah, it's actually available online. This would be like, I think it's interesting. A lot of our customers do that and just sort of compare it side by side. And of course, in developing that, we are like the first person kind of pioneered all this was Stephen Horvath, who, great mathematician, just took all kinds of methylation data sets and started looking through how different tissues are aging, and if he can predict the biological age or chronological age based on this methylation profiles. And I think a lot of scientists these days use that clock to in their experiments. And I really like these two lines of work, like measuring biological age based on blood biomarkers and the DNA methylation. They provide kind of two different pictures on the same process. And because DNA methylation is so tightly kind of bound into our physiology, because a lot of what we eat, what we do, how we exercise, what lifestyle do we lead is actually reflected in our basic metabolism, and basic metabolism influences DNA methylation. And DNA methylation is sort of like a really quick and dirty mechanism for the cell to regulate its activity. So all the methyl groups that get produced, it's like markers that cells very easily can put in the right places in the right time, quickly regulating up or down gene expression and therefore regulating this physiology.
So is
So
it fair? Excuse me, is it fair to say then the genetics our genetic code we get from our parents and our ancestors, but our epigenetic modifications through DNA methylation is one of the three types. We get from our lifetime experiences, our lifestyle choices and we could change that through our lifestyle.
in the way, yes, I believe we can. I think a lot of even methylation in part we do inherit, because it does write down, like, the body kind of already have an information of, for example, what your mom was doing during pregnancy or what your ancestors did in the previous generations. But I think that it's changes. It's much more fluid than genetics. Genetics is kind of a blueprint of your life, if I may use this an analogy. And epigenetics is something like add-ons that you putting in by living a life either in a healthy way or not a healthy way, or if you're doing right things or not so right things, I guess. And I think um, we can manipulate our epigenetics, and we can hopefully we can learn more and more about it. So it's a very young field. I would say we know very little. We don't know more than we know, but we start building this new biomarker. Said this like with epigenetics, clocks is one of them, but there are also panels for detecting diseases and looking at a different metabolic and physiological state. So I think it's just way in the beginning before like we really use it in a full capacity, I would say.
Yeah, and the methylation being affected by the grandparents. We just had Judith Finlayson on the program, who has written a book with a great title of "You Are What Your Grandparents Ate," but it basically deals with epigenetic modifications in utero passed on essentially 2 generations there. But
Yeah.
the the DNA methylation that we're talking about, how does that compare to other types of clocks for aging, such as telomeres we hear about, proteomics, glycanation? What are those measuring, and how do they compare with DNA methylation?
So I guess the most famous one is, of course, telomeres, just because they've been around for so long. And it's been shown like long time ago that telomeres are shortening as we grow old. And that was one of the hypotheses that when telomere shortening become very critical, that the whole, it triggers the process of the whole body kind of decomposition in some way, or the kind of destruction. Um, however, um, it's been shown that how to say this, it does correlate for sure, but at the same time, there was the processes that we clearly think that define aging process do not lead to telomere shortening. And one of them, I think I read an article somewhere that certain processes like diabetes, for example, we know that people with diabetes really like they need to control their sugar and level insulin levels and everything. And a lot of them were measuring their telomere length, and the telomere length was not predictive of the accelerated aging process, like for example. So I think the potential there was great, and it might be also depending on like what tissue you measure. So a lot of times the telomeres are measured in leukocytes and blood, and a lot of and maybe that's the problem. Maybe leukocytes telomere shortening is not really as conductive, like as doesn't demonstrate that widely how fast or how slowly you're aging. But I think the major problem here was, and this is why I'm not really criticizing it too much, and I really would like to actually compare it side by side. I believe that every time we develop a new clock, we don't do like comparative studies against all the other clocks that already exist. And I would really love to see that happening, because I feel like every clock has their own space in the field. Like for example, telomere shortening might show some aspect of cellular aging. And then Stanford was developed, as you mentioned, the biological clock based on a proteomics, when they took the blood from healthy individuals and of different ages, and they were trying to predict the biological age. So again, it requires a lot of work of mass spectrometry to identify all the proteins, but it looks very promising because it's based on exactly like a composition. It's kind of downstream of your genetics. It's what actually been produced, what proteins been produced, and in which form they exist in your body. And then another clock was based on microbiome, which we called the second brain of our bodies. And actually microbes do outnumber us 10 to 1. So it would be foolish to exclude them from the clocks. And there is different companies, Deep Longevity as well as Viome, developed their own biological clocks based on microbiome. And they also can predict biological age for quite great prediction based on what your microbial composition of your gut. And of course, biological clocks based on DNA methylation. So we have phenoage, I just talked about. Then there is a Horvath clock that some companies actually offering to their customers. So they're looking at about 356 sites on your DNA for methylation. There is Elysium Health just rolled out their panel. It's based on Beth Levine's clock that I was just talking about. It's phenoage. Also, they look at DNA methylation of over 1,000 fragments. And of course, science went even further, becoming really bold and trying to predict your mortality and also the day, kind of time to death with this Grimmage, I guess.
Grimmage, yes.
Yes, clock. And I apologize if I did not mention some other clocks here, because there is many of them. So I think consumers and scientists these days have great variety of clocks to work with and see which one is best. But I would really love to actually compare them all against each other and see the strong and weak points of each of them, so we can actually have make a very good decision of which one to use in which experiments and how.
So each of the clocks that we have available, and we have many different types and many different technologies, seem to look at a different aspect of aging, and they may reflect upstream effects or downstream effects of the aging, and that's going to be important when we apply them to ourselves or to research.
Yeah.
Now, sort of focusing on DNA methylation clocks in particular, the DNA methylation is a process which, when I first began learning about it, I thought of it just as kind of aging. It's sort of like rusting metal, acquiring more and more of these. But as I understand it now, it's much more nuanced, in the sense that net methylation doesn't necessarily change with aging, but rather just the locations of the methylation. So it's not that methylation per se is bad, but it's particularly the location, or bad, but I mean associated with increased age, but it's rather the specific locations of the methyl groups and the methylation that occurs on the DNA. Is that correct?
Yes, it's absolutely correct. Actually, methylation is very good for you. It's super useful. And we inherited through our ancestors a lot of this sites where a lot of methylation happen, and those sites of very repetitive sequences. And it's actually, they have to be protected by methylation from recombination or from there's a lot of transposable elements sitting there. So if they jump out and start modifying your genome, that was shown to be causing like a lot of age-related diseases like cancers. So we should not be thinking about a global methylation as a bad thing. Absolutely not. Yeah.
And in fact, removing methylation or winding back epigenetic effects, like using like Yamanaka factors or something to do that, will actually return the cells to a stem cell state, which is something will de-differentiate the cells, which is something we necessarily don't want to do. Although David Sinclair is doing some interesting work with the mouse model that he just published with the vision loss and then the OSK Yamanaka factors. But that's a whole nother area there.
Yeah.
So I've heard methylation clocks are referred to as like Steve Horvath's clock, his initial one, as a sort of first generation clock that was very analogous to chronological age, which, while it may have a lot of applications in forensics, for example, it's not really that valuable necessarily on longevity, because chronological age you can just get from your driver's license by looking at your birth date. But then the second generation clocks we're seeing now, sort of like Grim Age from his group also, and others, or are more valuable because they reflect biological markers of aging, and then more closely respond to interventions and everything as well. How does the clock that you developed fit into this whole thing, and how was it developed with your company?
Yeah, so when I started looking at the biological clocks, we had three main ideas in mind. So I did want a biological clock being very robust and predict your healthy age, let's put it this way, as accurately as possible. It should not be, and should be inexpensive to do, because we've been pretty much two scientists starting up the company with pretty much zero money. And so we wanted to make sure that we can do it ourselves and manage to process samples accurately. And it has to be very easy to collect, so painless for the customers to submit their sample. So saliva was the obvious choice from the beginning, because I don't know, well, you being a doctor, you probably, I don't know how much attention do you pay to it, but I know among my friends, they're dreading to go give blood tests. So
I hate needles too.
Yeah, they don't like needles. And for me, this test in the beginning, I did not think about this. No, I wanted to be as serious as possible, but I did not want people to go through stress of giving blood, because they knew they will not retest. And that was the point of following up your aging process throughout your life. Like every 4 to 6 months, you would measure your biological clock to see how well you're doing when you're changing diet, or you start new supplements that somebody recommended to you, or things like that. So the first thing we looked at is I pulled all the data sets that were associated with aging process. So there are gene expression that changes during normal aging. And we try to look as many markers as we possibly can, genes as we possibly can, just to see if we can get some kind of tissue, like cross-reactive, the same sets that are reacting similar across multiple tissues. So that presented a little bit of a problem, but we managed to settle on from publications, just comb a lot of publications, and found about 400 something genes that we settled on. And then, of course, there was a lot of methylation studies published by that day. So again, we pulled all the publicly available data sets. And now with a target in mind, I thought that if the methylation site would be relevant, we put another constraint. So that
were those, excuse me, were those 400 sites similar to the I think it was around 300 sites for Steve Horvath's initial clock?
I wasn't looking at that at the time, actually, because we wanted to be sort of independent. And the reason for that, why not looking, So at that time, I start seeing a lot of publications come in saying that the sites, what Steve used, he used very not controlled, I'm looking for the word here, sort of mathematically, he approached it from a mathematic kind of point of view. He just looked at everything and devised his model. And a lot of sites, and I heard it from a scientist who used this clock, a lot of sites landed in the dark areas of genome. So it's intergenic region that we do not know if they're associated with some genes or not, but they seem to be way in his clock in the predicting your age. So I wanted to be more biologically significant process. So that's why we started with age-regulated genes, and I didn't map it to his clock on purpose. Like I thought, okay, let us go through this exercise and see what we found. And so we put a constraint. So of course, every gene has multiple methylation site, we call this CPG islands, where the methylation actually happened and already been mapped. So we only selected those that were in the promoter region, because we wanted to see if it's actually regulating this gene expression or might be regulating gene expression. And I think in the first path, we also did maybe first axon, because sometimes regulatory elements within, like when within the first kind of part of the gene in the very beginning, shown to be the most important one. But that was a little stretch. We wanted to stay in the promoters, but we kind of laxed it just to include bigger data set. And then we looked through a lot of published data just to see if those sites, which of those sites changed the most across different population. And we had studies from all over the world. So we had a Korean study published, like we pulled in, and several study from Great Britain that they had also different nationalities of people submitted their samples. And that was the first kind of pass. So we identified about 29 sites that we wanted to pursue further. And very quickly we realized that 29 was not doable for a bootstrap company. So we settled on 9. That was really very doable for us at the moment. And we thought we're gonna, and we picked 9 that shown the biggest effect. So we thought, okay, we'll start with 9, test it on a healthy population, test it, and then roll it out as a first kind of minimal viable product, see how people do, how they test, and we will iterate, adjust, and then slowly introduce the rest of the clocks back in. So first we tested on the published data. It looked pretty promising. So we had our variation from chronological age of healthy individual that was from published data that was recruited through the hospital. So we assumed that those were seen by the doctors and declared healthy. Was about 4.5, 4.6 years deviation. So we're like, okay, that looks good. And then we start just calling friends and family, everybody who can spit in the tube, and and give us a sample. So we grown about 200 data points we got like that. Again, it looked very nice. So we thought, okay, maybe we should roll it out right now as a kit and see how it will behave with the wider population, where we don't know whether or not they've been healthy or sick or anything, and plan kind of adjust accordingly as we go. So that's where we kind of how we started. So now our test is 9 sites. It's based on saliva. Actually, by last count, our deviation was 3.8 years. So we improved our algorithm. Yeah, and hopefully we can. I'm very excited to see if we can start testing the rest of the sites soon and incorporate in our clock.
Yeah. Are there, or how many other sites would you like to incorporate ideally, or what would the rest of the sites look like? How many more is that?
So it's about, I think it's about 10 regions, and altogether the whole set would be about 32 sites. That's 32 methylation sites. So hopefully it's hopefully it's going to be easy to roll out, but we'll have to change our platform. We'll probably go more next generation sequencing, because that will not be able to accommodate that many people. Like testing that.
What's the time frame for that? Is that, how soon will that be happening, do you think?
Well, we hopefully will start testing hopefully in by the new year, and then as soon as we have a decent validation report. So I want to say spring, but I don't want to rush into it. I want it to be solid before we roll it out. So.
And just to be clear, the system is currently in use. I've actually tried it myself. Here's the package. It's something that you just provide saliva actually onto the paper. You know, it's not even a tube, and you mail that in. It's very convenient to use for that. Of the sites you're looking at now, are they more or less sensitive to any particular diseases or groups of diseases, or is it just sort of overall longevity?
No, it's overall longevity. Some of the, like I can tell you, like one of the regions that we're looking at is associated with integrins proteins, and integrins are apparently, we haven't looked at gene expression level yet, so we're planning on doing that. But integrins are a key part of our skin structural kind of proteins. They involved in wound healing and wrinkling of skin, and tied up in the whole like biological matrix kind of skin biological matrix, I guess, functions. So we are, the fact that one of our sites is actually might be regulating a skin aging is looks promising, especially after we try to be as unbiased in selection of the sites as possible. So that's like, I cannot disclose without the lawyers being after me exactly all the sites, but hopefully soon. But yeah, we did, they are, all the regions and all the sites are very much biologically active, relevant. They're not associated, as far as I know, with any diseases, although they might. I know for the fact that they do change the expression in different diseases. Like we briefly tested this set on, again, published data from cancer patients. And we did see like increase in biological agents for some cancers. We didn't test them for all, so I cannot claim everything yet. And I know that some of the patients did disclose that they have macular sclerosis. So I know that biologically they're much older that they should be, although they're managing it pretty well. So it's kind of, we're hopeful that it will work well, and if we expand it, it will work even better. So but again, we don't try to predict the disease, we just try to predict how well your body is doing at the moment.
So now we have clocks. We have a variety of clocks. In particular, we have epigenetic DNA methylation clocks, and we have a low-cost consumer version that's accessible to people. Do you think that these DNA methylation clocks will be, is it possible to reverse or lower the biological age as measured by these clocks through things like lifestyle interventions and such?
Well, apparently you can. We have a few very early adopters of this technology. So I want to mention this. When we first launched it, we were proposing it it was direct to consumer product, and we wanted to go direct to consumers, right? But we also did make a bid for a couple of companies that sell either supplements or some kind of nutritional products that's supposed to help you healthy aging. And we had a very difficult sell to the companies in the beginning, because the whole point of this was, you selling a supplement. I want everybody admits that there is no shoe that fits everybody, right? So there is some treatment will fit one person, and thus another treatment will not fit another person. So we offering your customers a kind of a window into their health. Is this working or not working? So they can give it a time, test it, and then if it doesn't work for them, quit and go and maybe change the supplementation, change their lifestyle, or do something else about their health. And it didn't go with the supplement companies. It didn't go very well in the beginning, and then we did made a very probably the best partnership ever with Ponce de Leon Health, and they were rolling out a supplement called Ridgewant. It's a calcium alpha ketoglutarate supplement, and they were really interested to actually offer they were so confident in their product that they were really interested in their customers to get this test. And at first, to answer your question, I actually did warn them. I said, I think in the best case scenario, we should expect not to see lifespan extension or reversal of biological age. I think in the best, what we will do, we'll see a slowing down aging clock, and your customers will not age at all. So they agreed to that we'll take that. And we rolled out the program. And surprisingly, so now it's been 2 years since we're partnering. And I can see that I do see that if the customers respond very well to their supplementation, we do see reversal of their biological clock. So we do see that, they did test by other biomarkers. So they did measure their some blood biomarkers, and they do see the improvements in overall health when they're taking alpha ketoglutarate. So that was at least I can see that the clock is working. So we were predicting their biological age and the starts, and it looks like the biological clock does correlate with some other blood biomarkers in at least those customers. And we also did like comparison with Horvath clock, with the other company that's selling it, more expensive clock. Unfortunately, we could only afford like 10 samples, but we correlate pretty well with another clocks as well. So we usually predict a little older biological age than Horvath clock does. But I'm actually right now in a couple of our current customers do measure using our clocks and Horvath clocks, and they see the trend is similar. So it makes me feel better that we're in agreement with the kind of a gold standards out there. So yeah, and I think since the Ponce de Leon, the more and more companies are enrolling in this program, and I'm actually very excited about that, because I think they're providing great value for their customers by showing that, look, we are actually not just sell you a product, we're actually genuinely thinking that you should know how well you're doing by taking it, and find your best therapy for aging.
Yeah, it's exciting that you're finding you can slow down the biological clock with these interventions, or even, some people even reporting reversing it.
Yeah.
Over the 2 years, did the effects slow down at all? Because obviously when you get to reversing your biological clock, I'll reverse it to, you don't want to reverse it too far. I'll take 40, I'll take 30. I'll take 20. I don't necessarily want to go back to 15 or,
Me
and of course
neither.
that's not really possible, but it's interesting when you talk about reversing biological age. Does it slow down? Have you noticed it in that 2-year period? Were you
Yes,
able
it
to
does.
see that effect at all? And
Yes, it does. The customers that are taking, I don't know if they disappointed as much as uh, I see, like initially they start doing something. If it works, it drops very fast. Let's say by 5, 6, sometimes over 10 years, and then it stays there. And I know some of them try to even lower it further, and it doesn't work. And it's again, I think it's super interesting. Why not? Why, if you lower your biological age to, let's say, 46, you cannot get any younger than that? So is it your limit, or maybe it's like this healthy steady state that we should be focusing on maintaining rather than continuing lowering down a biological clock?
yeah,
So
that's a great thought. The idea of a sort of a steady state that we return to, but necessarily we don't ideally roll it back continually. And then, like you touched on, a very key question: if we reverse the clock or slow down the clock, does that truly affect any underlying aging process? Because I'm broadcasting here from Southern California, where people have been reversing their biological phenotypical clocks many years. I mean, many times, and things like, dyeing their hair, their gray hair black, makes them look younger by that clock, or Botox injections for wrinkles makes their skin look younger, but at a fundamental level, it doesn't really change anything. But it appears there's growing evidence, or there's evidence accumulating that reversing these DNA methylation and other physiological clocks can actually reflect significant things about effects on longevity.
yeah, I heard, well, of course, the growing number of published data showing that. Also, people recently, for example, was a paper published that certain diets, like for example, rich in polyphenols, people went on their diets, start exercising, start changing something in their general lifestyle and behavior, and they did get biologically younger, but they also provided some other biomarkers where they're measuring other aspects of their aging, and they also saw improvement. So this is actually where we're going. Hopefully we will go next, and I'd really like to do more like sort of clinical trial study, like whether or not if we take so for example, in the cohort that we already have, when we take a person who self-reported poor general health, start doing some intervention, whether or not they're taking artificial glutarate, or they went for a diet, certain diet, or they did intermediate fasting and or something else, and they saw a dramatic improvement, and we look at their biological clock dropped down more than 12 years. Actually, we do have even policy of we automatically retest you if you tested too young or too old, just to make sure that we actually showing you your true biological age. And then this person over sun reports that they're feeling good. I know it's subjective. I understand that it's just their feeling, how they evaluate their biological health, right? But it's one of the metrics, and I feel like of course there is an effect of placebo. You cannot get away from it, but I think if the person really feels better, that's a good indication that something might be working in the right direction.
Yes, and we've interviewed on this program Dr. Lucia Ranica from Stanford, who is working with low carb diets and some type of fasting to show reversal of DNA methylation epigenetic clocks, and also another clinician on the East Coast, Kara Fitzgerald, is enrolled patients in her study, and it involved dietary changes, typically ketogenic diets or low carb diets, and then fasting, as well as her program had exercise and some sleep things too. But she showed over 3 years reversal of epigenetic age as measured by the clock they used. They were using a Grimage type clock,
I see.
but that was only after 8 weeks of intervention in their patients, and that was a randomized controlled trial. It is a pilot study of only 40 patients, I believe, but at least it shows the possibilities of what this technology can do.
Absolutely, yeah.
So it's particularly great that now individuals can get access to DNA methylation clocks through companies like yours for, affordable prices if they want to begin following their own lifestyle there. And speaking of lifestyle, now with all your expertise in DNA methylation and genetics and epigenetics, and how has this informed your own personal lifestyle choices, Yelena, for how you live your life?
Actually quite a bit, surprisingly, because, well, I'm got full disclosure, I guess. I've been struggling all my life with obesity, and it's genetic. I'm going against my genes here. And I have to say that at some point you do buy into all this diet and hype, I guess. And when we developed the test, this was the first thing I decided to test. So I recruited several of my friends who are in different kind of body composition, and we decided to test the ketogenic diet. I was determined that seeing all these people, not just slimming down and losing weight, but also claiming to improve conditions, like chronic conditions that they developed, I thought it must work. It, like, look at this. This is such a great body of evidence. Like, I should try it. And again, I have to say, like, I'm clinically, I'm absolutely healthy person. So I decided to go anyway on that diet. So obesity being the only issue here. And I measured my biological age before and after, after about actually 12 weeks, first measurement was. And so in the beginning, I was about 2 years older than my biological age. And I was like, well, makes sense. Well, probably I'm as old as I am. So obesity did not affect my age quite yet. So I went on 12-week diet and measured again, and I become 7 years older. And I started thinking about, I didn't quit the diet quite yet. I thought, okay, I lost so much weight, and it might probably ignited some stress response. So I started digging into what other biomarkers I could measure to see if my biological age was indeed much older. So I actually turned out to be, so in that point in my life, I was also very much interested in microbiome, and I still am, but then I actually worked for a company that were doing microbiome testing. So I decided to test my microbiome, and my diversity went down quite a bit. And I tested with a different company, and that point was very young company, Biome. Right now they are probably one of the best in the market, and I use them a lot. And the same story, they showed me that my diversity on microflora goes down dramatically. So I tried ketogenic plant-based diet. I become even 2 years older than that. And diversity improved a little bit, but again, it was not as good as it was before. So I quit that nonsense and went back to like more plant-based diet, and I practice start doing intermediate fasting. Well, I probably not fast long enough to impact my weight as much, but definitely my health improved quite a bit, and my diversity of my microbiome improved a lot. And I'm right now actually 5 years younger than my biological age, surprisingly.
Primarily
So
from the fasting, not diet
yes,
per se, just controlling when you eat, not what you eat. Is that right?
well, I do not eat that much of red meat, although it's hard for me. I guess I'm also, this is another thing. A lot of people just jump into vegetarian kind of lifestyle. Again, measure your biomarkers. You can find out you cannot, like I cannot be vegetarian. I cannot, for some reason, absorb iron from anything but red meat. But eating red meat maybe once a month is more than sufficient for me to keep my healthy iron levels. But yeah, I'm trying to be more like Mediterranean type of diet, so more fish, vegetables, less meat. Um, but yeah, I try not to stress too much what I eat. Just eat good food, cook as much as I can at home. Um, and then I just practice intermediate fasting. So that's kind of what I do.
Uh-huh. Any particular supplements or drugs that you recommend?
Um, so I do again, I try to satisfy my second brain. So I do follow right now all the recommendation that Biome give me just to test it out and see how it works. I experiment all the time actually on that. And I do take alpha ketoglutarate, so I start taking that as well, just because I wanted to. We see such great results that I wanted to test it on myself. So that's the only two things I'm doing.
Uh, with your interest in longevity, so no NAD supplements or resveratrol or metformin or rapamycin.
No,
It
no,
sounds like what you're doing is working for you
yeah.
as it is. That's great. Yeah. What has been the what's the most impactful thing you've done, do you think, to improve your health overall with your lifestyle?
Well, this was surprising that this combination of looking after your kind of second brain and looking after and doing intermediate fasting so far
Hmm.
has been really impactful. I still need to measure the effect of alpha ketoglutarate in my health. Like it's still coming up. I just I did it before, but I recently kind of introduced it again into my regimen. I feel that staying positive, also having an exciting life, and like for me, it was developing something new and um, kind of using my brain also to kind of develop new tests or new ways of where we're going with this empowering people. I think it's kind of gives me a purpose in life and also satisfy my social kind of circle and social interaction. I feel like we maybe focus sometimes a little bit too much on what to do to extend our healthy years and with pharmaceuticals. I think we should more like listen to your body, listen to our body, and kind of listen to what it tells you and go with that. So that's what I don't know. I think this is when I stop worrying too much about like excess weight or stress at work or something like that and just do what I enjoy. I think my lifestyle improved quite a bit.
That's great advice. How can people follow you on social media, or can you give us the website of your company?
So yes, the website is trumelabs.com. And without www in the beginning, just trumelabs.com. And we also have a Trume Labs group on Facebook and Instagram, where we're trying to find, just right now, publish new things that we find that our customers sharing with us on what's affecting aging. and how well they feel, or maybe current studies. I'm trying to report on that as well. I should be more active on social media, so for sure, but we're very responsive. So if you have any questions, we will answer any questions you get either through our website or through social media channels. So yeah, and hopefully more will come soon. So we're just growing our team. And I hope that as a scientist we'll grow more products and go to next level. The marketing team would actually be reporting on that and keeping our customers up to date.
Well, thank you so much, Yelena. It was great to be able to spend an hour with you here today and hear about the exciting work you're doing and the work that you're doing with your company as well. And I look forward to staying in touch and hearing about the new developments coming in the new year as well.
Yes, absolutely. Thank you so much for inviting me. It was very nice to talk to you about it. Yeah.
Note, this is not intended to be a substitute for professional medical advice, diagnosis, or treatment. Always seek the advice of your physician or other qualified health provider with any questions you may have. Never disregard professional medical advice or delay in seeking of it because of something you have seen here. If you find this to be of value of you, please hit that like button and subscribe to support the work we do on this channel. Also, we take your suggestions and advice very seriously. Please let us know what you'd like to see on this channel. Thanks for watching, and we'll hope to see you next time.